Today is my great pleasure to have Dr. Lu to join us to deliver today's webinar. Here is a brief intro of Dr. Lu. Dr. Lu is the co-founder and CSO of TriArm Therapeutic Corporation, a company specialized in developing innovative cellular immunotherapies for unmet medical needs. Prior to joining TriArm, Dr. Lu worked at the US FDA as an expert reviewer at OTAT, Office of Tissues and Advanced Therapies, and CBER, the Center for Biologics Evaluation and Research. Dr. Lu is a recognized expert in the field of cell and gene therapy and has co-authored several FDA guidance publications for industry leaders and practices. Dr. Lu served on the FDA's Scientific Committee of Oncology Center of Excellence and was the recipient of the CBER Award for Excellence in Regulatory Science Research in 2015. Prior to joining FDA, Dr. Lu worked in the biotech industry as a scientist, senior scientist, manager, and consultant in the development of viral vaccines and viral vectors for cancer therapy. Today, Dr. Lu will share with us his years of experience in the field of immunotherapeutic research and development, from science to regulatory and back to R&D. So the title of Dr. Lu's talk is Preclinical Evaluation of CAR-T Immunotherapies using Novel Animal Models. Dr. Lu, great pleasure to have you today. Without further ado, the floor is yours.
Thank you, Dr. Lee, and it's my pleasure for have this opportunity to discuss with you all about this topic, which I consider is most critical for developing a beta CAR-T product. So let's start. Do everyone hear me well?
Yes, very well.
Good. So here's the outline of today's presentation. I will first have a brief introduction about the CAR-T product, and then discuss issues the field's current phase, and the challenges moving to the next stages. The utility of animal model in these steps will be discussed in the context of overall preclinical evaluation of CAR-T products, and then give a brief summary. So here is the introduction of CAR-T immunotherapies. As we all know now, the CAR-T is very hot. Here's the Science magazine's cover story. In the year 2013, it was denoted as breakthrough of the year. And the chimeric CAR T cells stands for chimeric antigen receptor, CAR T cells. It's together with the immune checkpoint antibody is most promising categories of the immunotherapy we are developing. Even though the CAR is relatively new to most people, but actually technology has been existed for more than 20 years already. So what is a CAR? This picture depicts the CAR product's mechanism of action. On the right, we have, let me have the pointers here. Here we have a cancer cells. Typically, cancer cells sometimes will express unique or differentiated expressed markers we call the tumor-associated antigen, or TAA. Many times, TAA will be presented associated together with MHC complex and are recognized by T cells on the surface by the T cell receptor we call TCR. However, the scientists developed a new way for the T cell to recognize the cancer cells and develop this so-called CAR technology. The CAR, as we mentioned already, is a chimeric antigen receptor, typically have antigen binding moieties and then linked together with the other segments. And once you have the TA and the CAR interacted, the T cell become activated and then release lots of the cytokines, chemokines, and also the toxins to the cancer cells. And the cancer cells be killed. Oh, I see. Sorry, I cannot just change easily. So let's take a look further on the CAR designs. On the left, we see normally the T cells express TCR and recognize tumor antigen in the peptide and MHC complex. And the CAR to a degree is structurally similar to TCR and MHC peptide complex. By that, we mean we have antigen binding moieties typically comes from a single chain fragment, variable fragment of antibody, which can recognize tumor antigens. And then we have a linked sequences connected to a transmembrane domain, and then followed with the intracellular domain. Intracellular domain typically have a signaling transduction function. And for the first generation, the signaling transduction domain only comes from the CD3 complex zeta chains. And then the clinical outcome from the first generation did not yield success. So the scientists further made the further improvements and designed the second generation CAR molecules. And for the second generation, we have two signaling transdomains, either from CD28 or 4-1BB molecules, together with the CD3 zeta chain. And a further improvement has been also generated. We call the third generation, which contains two signaling transduction domains from the CD28 and 4-1BB or other activating molecules. And we also have the further innovations to generating so-called fourth generation CARs. And in this case, once CAR molecules be activated, it will stimulate the production of the certain cytokines such as L8, L2, or other cytokines to further enhance the anti-tumor activity. The production of the CAR is quite complicated. First, the cells will collected from the patient, and then the T cells will be selected and activated by the cytokine or antibodies. T cells will further infected with the viral vectors, such as gamma retroviruses or lentiviruses, which encoding are CAR molecules. And the cells, infected cells will further expanded and then go through quality control testing. And the cells will then infuse back to the patient. Before the cell infusion, typically the patient will receive a preconditioning with chemical. So the CAR product is quite complicated. So why scientists want to develop such a product? We already have the antibodies, we already have the vaccines, and others, even the stem cell transplantation, which in general produce the good results. But in truth, the CAR have several advantages. First of all, it utilizes the T cells, which is the major players for anti-tumor activity. Second, to a degree, the CAR is the first time people realize the dream about designing immunity. For example, in this case, we can choose the antigen we want to target it to. Second, we can have a much better binding affinity than the natural occurring T cell receptors. Our T cell typically have the affinity, it's hundreds or thousands for the less than antibody binding. And also, the CAR is insensitive to some tumor escaping mechanism. For example, the tumor cells will reduce the production of the expression of MHC, or even have the defect in the antigen presenting stages. So therefore, the tumor cells have less tumor antigen presented on the surfaces and normally will not be recognized by the T cells. However, the CAR is not dependent on the MHC presentation. And also, as far as you have any cell markers which is associated with the tumor cells, which potentially be recognized by the CAR cell. And we can also modulate the strength, types, and the duration of the immune response. For example, after we infuse the CAR T cells into the patients, if we do not want to have those CAR T cells anymore, we can wipe out with the suicide or other mechanism. And also we can, by adjusting the cell numbers and the cell types, we can tilt the immune response to, say, immune suppressive or more enhanced immune response to it. More importantly, by expanding and activating the tumor cells, the T cells ex vivo, we avoided immune suppressive environment, which in generally is the case for the patient with the tumors. So how does CAR perform clinically? Here is the result from the BLA, the Novartis CAR T cells. It compared to bispecific antibody, also compared to a chemical. We can see with us from the overall response rate, which is dramatically higher than the chemical drug and also antibody. And from the median overall survival, it's also dramatically longer than the chemical drugs or the antibodies. So taken together, the CAR T cell actually the quite excellent result clinically. In the right panel, here's the result from the long-term follow-up. In this case, the CD19 CAR was used to treat young adults with the B cells acute leukocytic leukemia, BALL. And then we can see the plateaus of the survival, which is typical of immune response, the immune therapies. And the survival could be last more than 5 years and still have quite significant portion of the patients, sometimes more than half, 50% of the patients to survive. That's achieved the theoretical cure of the cancer. It is exactly due to this kind of like excellent clinical outcome, the FDA first approved the 2 CAR drugs in year 2017, and in the following years, additional 5 products had been approved for various indications. And also the CAR T had been approved by the Japan Health Authority. 2 CAR T products had been approved by the Chinese regulatory agency, which is jointly developed by the domestic partner and also the partner from the US. So now we move to the challenges in developing new CAR T therapies. Even though the currently CAR T cells achieve the greatest success, it also faces several challenges. First of all, right now, the approved drug is only for the blood tumors. And we don't have the drug for the solid tumor yet, and not for the other indications such as infectious disease or the immune disease. Second, we already have that the drug resistance occurring. For example, anti-CD19s, we have the mechanism, we have the tumors escape the CAR T cells by mutations in CD19 mutations or the modification of the CD19 molecules. And so the cost of the CAR T is quite expensive, ranging from $200,000 to $500,000, $200,000 to $500,000 US, which made it not so affordable to most people. And so far, even though after years' efforts, still the quality of the CAR T have the rooms to further improvement. That means the consistency of the product and also the safety and efficacy. Lastly, the speed of manufacturing is still the issue. Currently, the base production process need at least 1 or 2 weeks of manufacturing time, and many times some patients will not have to afford to waiting for such a long time before the cells infuse back to the body. So what are some of the target goals we try to achieve in order to have a better CAR-T product? First of all, we try to have a better smart CAR construct, such as the CAR activation is based on the logical design. And also maybe have a modified effector mechanism, not only depend on the natural T cells effector mechanism, but some artificial ones such as the toxins. And another directions very hotly pursued is the third-party off-the-shelf CAR-T products. In that case, the CAR-T cells can be more timely infused back to the patients and also could potentially achieve the cost effective. Another one is the try to express CAR in the stem cells. In that case, the cells can be more, can exist in body longer time, so achieve a better outcome to prevent relapse of the cancer cells. Another direction is combination of the CAR-T cell products together with other cell therapeutic modalities such as oncolytic viruses or chemicals or antibody therapies. As you can see, there are hundreds of the companies are trying to develop different kinds of the technology platforms. For example, they are using different carrier cells like NK cells or macrophages, and they are expressed that the CARs in the allogeneic cells, which to make so-called universal CAR-T cells. However, greater challenges are ahead. In these reviews, they also summarize some key issues to develop the better cancer immunotherapies. On the top is the development of preclinical models that translated to human immunity. That puts the novel animal model development into a very prominent space to further improve that CAR-T product. If you look down this list, there are many aspects also require beta animal models. For example, this is one of the response I received from the sponsor when I reviewed one of the applications. The response basically stated, in order to have an animal model to mimicking the human CAR-T cells in vivo, they met so many different requirements. And unfortunately, such kind of requirement cannot be fitted by the current animal model. So the sponsor argued we should try to use the alternative way to evaluate the CAR-T products. This also case clearly illustrated the difficulties to have a better and novel animal models for the CAR-T product evaluation. So next, I will move to develop the animal models in developing the CAR-T therapies. Before we discuss the animal model development and the utility in CAR-T therapy evaluation, let's look back what the preclinical study are for. To conduct the preclinical studies, we basically try to establish a scientific basis for conducting clinical trials. First of all, it's feasible to conduct such kind of study. Second, we want to establish a proof of concept. Such kind of study is rational, is reasonable. And we also try to provide some hint how to design the clinical trials, such as identify the patients, identify the possible starting doses and dosing schedules, and identify some issues which could be treated or prevented during the clinical trials, or even just to find out some parameters for clinical monitoring in order to look at the product safety and product efficacy. And lastly, many times the regular agency will require such kind of studies, so we need to conduct this study to meet these requirements. Since the gene therapy and cell therapy is very hot, so many guidances, many agencies are busy to issue various directions guidances, either from the ISH, from the US FDA, and from the Chinese regulatory agency, or even from the EMA, just for example. For example, in this case, the guidance issued by the US FDA year 2013, which is the first guidance for the CAR and also for the cell gene therapy product evaluation. And at the same time, the ICHS6 document is also mentioned when considering the preclinical evaluation of the CGT product. Since my working experiences and background, I will focus on the regulations from the US and also the practice from the US. This slide basically summarizes the essence of regulation we are using for the cell gene therapy products. First of all, there are no one-size-fits-all regulatory approach. So we always take a case-by-case approach to evaluating and to set the requirement for the different products. So in this case, the data to support the clinical trials is in related to the product itself and in relation to the specific clinical indications. And when we make decisions, it's always based on the balanced risking and potential benefit. Just a couple months ago, FDA issued a draft guidance. It's more specifically tailored to the CAR-T product. This one is for the CMC and also preclinical. The framework for the preclinical evaluation of CAR-Ts is summarized in these slides. First of all, the product safety is not just the look at the product itself. It could be the components used for generating such product, and it could also from the manufacturing process or the biological activity of the product. Safety concerns could be from the individual factors or the combination of the interactions of different factors. I will have the examples to illustrate these points. In the CAR-Ts, this aspect could need to be evaluated, such as tumor-associated antigen itself, cells used, antibody used for designed the binding moieties, the vector used, and also the systemic toxicity from the product, and also the regimen-related toxicity, such as the chemical treatment, resulting in certain safety issues. Here is an example for the so-called TAAs and how to testing. The tumor-associated antigen could be unique to the tumor cells, and it could also be just a restriction to tumor and certain tissues, such as cancer testis antigen. And this TAA could also be due to the differential expression, such as have the high level expression on the cancer cells, but not in the normal tissues. Or it could be highly expressed at a certain stage or tissues at the development stages, but then later on, it it was disappeared or reduced, but still maintain a high level expression in the tumor cells. Depending on different situations, different testing strategies can be explored. And first, we need to consider previous experience with the target. For example, like a MARC1 or EGFR2, 3, they have lots of the different clinical development based on, say, vaccines, antibodies, or other strategies. Those clinical experience with concerning the safeties can be very useful when we develop the CAR-T product. And if it's had homologous to the targets in animals, then we can assess the TA expression in animal, have better pictures how it was expressed, even in some rare cases, very, very low level of expression in certain critical organs or tissues. And we certainly need to assess the TA expression profiles in different tissues by using the tissue cross-reactivity assay or protein arrays or tissue arrays. And if necessary, we needed to conduct in vivo studies. For the vectors, we have many vectors can be used to express CARs, such as the retroviruses, transposons, nucleic mRNAs. And their safety concerns come from the insertion mutagenesis set by the retroviruses or lentiviruses or by the transposons. And sometimes those viral vectors can produce the replication candidate viral vectors. It need to be assessed. And this study can be done by in vitro or in vivo. The cells. CAR-T cells, even though have the T there, but as we mentioned earlier, can have different varieties. It could be T cells, even T cells, you can have the subpopulation of the T cells, or T cells are the different stages of the development stages. And it can also have the other cells such as NK cells. So safety evaluation need to consider the uncontrolled proliferation potential and a karyotypical analysis to see if are there any changes due to the genetic manipulation. Are there any clonality due to that in vitro ex vivo expansion? And also, if the T cells is antigen specific, we need to demonstrate in vivo or ex vivo to show this antigen stimulation still works. So what are some of the model systems used for the CAR T cells evaluation? We have in vitro models. In vitro models have a variety formats, such as expression, the target cell lines, tumor cell lines. It could be tumor-derived organoids, and it could be stem cells or panel of the primary cell lines. And by using the primary cell lines, we can be have more sensitivity to detect minor reactions due to that CAR interaction or recognition of the low expression antigens. And we can use the protein or tissue arrays. And we also have the in silico models. In this case, there are lots of the in silico analysis tools to analyze the databases to demonstrate antigen expression profiles and also identify potential off-target issues. And we have the in vivo models. For the in vivo animal models, we also have different choices. We have the immune competent models, and in this case, it's suitable for syngeneic tumor models, and we can start the preconditioned regimen evaluation in these animal models. And certainly we can accessing the CAR-induced host immunity or study the targeting suppressive tumor microenvironment. We can also use the transgenic mice models. For example, use the transgenic mice express the human HLAs to start the HLA restrictions or identify the antigens restricted by the specific HLA molecules. And we can assess in vivo CAR generations and to assess its activities. Most commonly used is immune-compromised models. And this one can be used to assess the many aspects of CAR-T products, such as the pharmacology and the beta refinement of the CAR constructs, trafficking, proliferation, and adverse effects. And currently, very promising fields is developing humanized mice models. In this case, the human immune system is partially reconstituted in the immune-compromised mice, and then have a beta environment to assess CAR-T products. Look at when should we use these animal studies? In this newly issued guidances, the FDA identified several occasions the animal study can potentially be useful. For example, to using the assessing targeting specificity, to assessing antigen-dependent and independent activity of CAR-T cells, to evaluate the T-cell clonality and cell trafficking and proliferation. But before we jump into that efforts, we need to bear in mind there are lots of limitations to use animal models. That's exactly due to the species specificity of the cells and the antigens, and also other difficulties in modern human immune systems in animal model. So it is true that several animal models is quite useful. We're generating useful information to assess CAR-T cells. However, there are lots of limitations, particularly because CAR-T cells is a live drug. Many times, the selection of starting dose level is not determined based on solely on animal studies. And the previous clinical experience with similar CAR-T products can often inform starting dose level, dose escalation plan, and a dose regimen in the study population, which is quite different from other drug categories. So talk about that animal model. What do we have here? And here I'm just to focus on the mice. So then, first of all, we have the normal mice with a complete immune system. By that it means we have all kinds of the immune cells in place. And then we have the neuter mice, which is lack of the T cells. And we have the not skid mice. And in this case, the TB cells disappeared. And furthermore, we have severe immune deficient mice, NCG strains, NOG strains, NS strains. In this case, T cells, B cells, the NK cell all disappeared, only had the residual macrophage and the dendritic cells. For example, in this case, it's NCG strains. NCG strains is a severe immunodeficient mice model. It have a knockout of the two key genes, which is critical to regulating that immune response or immune system development. And by the flow cytometry analysis, you can see in the NCG mice, both T cells and B cells and NK cells are absent in this cell, in this mice. In order to have a humanized mice model, we need to first look at what's the differentiation pathway for the hematopoietic stem cells. In this, hematopoietic cells were differentiated into a common myeloid progenitor cells and lymphoid progenitor cells. For the myeloid progenitor cells, we were further differentiated into monocytes, macrophages, and for the lymphoid progenitor cells were further developed into the B cells, T cells, and NK cells. And then by infuse PBMC back into the immune deficient mice, we have a partially reconstituted certain immune functions. And if we infuse back the human HSC cells into the NCG mice, we basically will have all kinds of the human immune cells developed in the NCG cells, therefore offer the better opportunity to study the effects of the human immune systems to the CAR T cells. In the next few slides, I will just use some examples to illustrate the utility of the animal models. So in this case, the NCG mice was transplanted with the tumor cell line NAM6, which expressed the luciferase, and then infused with the CAR T cells. In this case, had two versions of the CAR T cells. And then we monitoring the anti-tumor activities by looking at the bioluminescence levels. And as shown depicted in these figures, if the mice infused with the control T cells, its tumor grows quickly. However, with the infusion of the CAR T cells, then the tumor growth was completely controlled by the CAR T cells. Therefore, the NCG mice in this case demonstrated is a good animal model to evaluate the pharmacology of the CAR T cells. CAR T cell is a gene therapy product and many times involved at the viral vector transduction. And we already know the viral vector transduction into the stem cells many times is tumorigenic. In this case, in this review, I reviewed many cases of the viral vector manipulated stem cells, and sometimes they will have the tumor developed in those patients. Therefore, proper assay needed is required to assess the product tumorigenicity potential. In this case, it's NOG mice were utilized to assess the tumorigenicity of induced PSC cells. And you can see in this case, the NOG mice is very sensitive to the tumor formation. With HeLa cells, with about 10 cells, you can have 50% of the mice develop the tumors. However, for the iPSC cells, you need about 100 cells to develop the tumors in the 50% of the mice. For the current approved CAR T cells, this kind of assessment is not required yet. However, with the newly developed CAR T cells, many times people try to have a better persistence of CAR T cells. They will utilize cells which is more stem cell-like. In that case, it's very likely such kind of testing may be required in order to evaluate the tumorigenicity potential of this kind of product. The animal model can also be utilized to assessing the potential of target safety issues. In this case, the researchers want to develop the CAR targeting the CD33 or CD123, which is the marker for the AML. So the NASG mice was infused with AML cells and then infused with the CIK cells express CAR against the CD33 or CAR or CD123. And after that, the AML cells remaining in the animals were sorted out, were infused back into the another animals and looking for the resistance to the CAR T cells. We can see in this case, the CIK anti-CD3 CAR cells have a great anti-tumor activities. And as the same for the CIK expressed CAR CD123 CARs. And in the animals, we can see these CAR cells expressed it exists at high levels. When the tumor cells subject in the first round of the CAR T cell treatment were infused back into that immune deficient mice, and then infused CAR T cells again, we can see these CARs again have controlled the tumor growth, both. And these CARs can express at relatively good level in the immune deficient mice. However, when CD34 hematopoietic stem cells infused back to the mice, and they later to differentiated, then infused with the CIK CAR cells. And then if we compare the colony formation, we can see CD33 CAR cells suppress the colony formation more dramatically compared to the CD123 CARs. It's the same if you compare the colony formation by the mononuclear cells. Therefore, CD33 CAR may be a less good CAR product compared to CD123 CAR products. In this case, the animal models reconstituted with the PBMC was used to study the cell-cell interactions. In this case, it's the Treg versus CAR. So a Treg cells will suppress the CAR T cell anti-tumor activities and let the tumor to grow. And if the mice is infused with a CAR expressed regular CD28 wild type CAR molecules, it has suppressed the tumor growth. But if we infuse the Treg cells together with these CARs, then we can see the anti-tumor activity was abolished and the tumor grows back. And we can see in this mice, and the Treg cells grows to more amounts. But if we make mutations in the CD28 domains, have a new CAR construct. In this case, again, if we infuse into the mice, the tumor growth will be controlled. But dramatically different from the previous version of the CARs. In this case, addition of the Tregs did not abolish the CAR T anti-tumor activities. And also the Treg cells did not expand. Therefore, CD28 mutations can suppress the Treg cell expand, the proliferation in vivo, which is quite relevant to the clinical setting, because in the clinical settings, we will have the Treg cells in a tumor microenvironment. The potentially CD28 mutations is a big product, and currently we are evaluating this product. The last case derived from one of the applications I reviewed. In this case, the researchers want to develop CAR Ts derived from the stem cells. They transduce stem cells with lentiviruses express CARs, and also the HSV TK genes. And then they want to evaluate the anti-tumor activity in the NST mice, and also to see if a chemical drug against clover, in this case, can deplete CAR T cells from the mice in vivo. The study was conducted this way. The cells were first, stem cells were first sold and stimulated, and then transduced with the lentiviral vectors encoding CAR molecules, and they injected into the mice. And then at certain stages, the tumor cells were infused back into the mice too. And then anti-tumor activity was evaluated by looking for the mice survival. We can see if the mice infused with the no transduced stem cells, then the mice die early. But if we infused with the CD34 cells transduced with the CD19 CAR or CD19 CAR together with HSV and TK genes, then the tumor survived. And in this case, it seems the CAR co-expression CD19 CAR and also HSV TKs have a better production compared to CD19 CAR only. Important here is this. If we treated with the Gansk-Garover drug or compared to the no treatment, in this case, CD45 cells in the bone marrow or spleens was not affected. However, in the bone marrow, the CD19 CAR cells was not affected. But if the CAR also expressed HSV TKs, we can see the CAR cells was depleted in the bone marrows, both by that CAR positive cells and also by detecting the vector copies. It's the same thing. The same thing happened in the spleens. Treatment against Garover depleted the CAR T cells only when CAR encoding both CD19 CAR and also HSV TK genes, but not the CAR only. Therefore, in this case, the mice model was used to study combination treatment. In summary, the CAR T fields have achieved great success, but still face many challenges to develop better products for both indications. Preclinical models are urgently needed for CAR T evaluation and direct product development. Certain novel animal models had been made, and greater progress has been achieved. But we need to keep in mind that there are many limitations when using the animal model for the CAR T cell evaluations. Therefore, we need to have a very early interaction with the regulatory agency and follow the guidance documents when we're designing preclinical evaluation programs for the CAR T products. Here are just a few useful guidances for. Thank you. That's the end of my presentation.